Today we are going to learn about how to visualize your data, how to visualize your docking results, and make a publication quality figure in order to publish some high quality papers. So I'm going to show you the whole process of analyzing the data from the docking results to a publication quality figures, and how to plot your data by using different softwares. So let's get started. So the first thing I'm going to show you is that this particular paper, which I published in 2020 during the pandemic, and it was published in Science Advances, and the reason why this paper has been widely cited is because of the quality of the figures and the data analysis that I have done by using different softwares, by using different free softwares. And these are some of the visuals that you can present your data in publications, in different types of publications, and you can publish some high impact papers as well. Of course, the analysis you require has to be some has to gain some insights from the analysis, but I think you have to also represent your data in such a way that even the layman can understand a little bit about the work that you have done and the purpose of the work. That is what is more important. Of course, you have to write a description about your analysis. You have to specify all the analysis tools and what are the insights from those analysis you have to present, but also the figures are really important as well. So let me show you some of the softwares in which you can analyze these docking results and you can present these type of quality figures. Okay, so let me close this and we'll go to the Vina folder, which I had shown in my previous videos, how to do the blind docking, how to do the targeted docking, and what are the files that you require, starting from the protein preparation to the ligand preparation and to the Autodoc Vina results. Okay, so let's get started with one of the approaches that we have done, that is called blind docking. And as I have shown before, that you can generate number of binding modes by using Vina_split, and these particular files which are generated from the docking results are called PDBQT files. These PDBQT files can only be opened by using Discovery Studio Visualizer and UCSF ChimeraX, not the UCSF Chimera. So if you try to open this by using Chimera, let me see, all software, and if you want to open by using Chimera, you won't be able to open that. There will be a type of selection that you want to click on that, and you have to modify different sections in order to visualize PDBQT file. But in my experience, you cannot open PDBQT files inside UCSF Chimera. But one good thing is, now UCSF has introduced a new software called UCSF ChimeraX. So if you want to open this PDBQT file, you can open it by using ChimeraX software. Previously, many people, where UCSF Chimera was not introduced, PyMol has been widely used, and PyMol can open PDBQT files. But due to the recent announcement of ChimeraX, you can do a lot more analysis by using this software. So let's open using ChimeraX. You can see that you will find these drug molecules, the PDBQT file. I think I have shown in my last video how to open this PDBQT file by using ChimeraX. So this is just the one pose of the docking results. If you want to open everything, then you have to click the output file from the docking widgets, that is called this one, 6LU7 drug.pdbqt. So if you open this by using ChimeraX, you will find the all 20 different poses, starting from it. If you expand this particular model section, you will see that you have all the 20 modes that have been loaded in the UCSF ChimeraX. So if you select, this is the select option. If you select, then this will be highlighted in green. So this is the first pose, this is the second pose here, this is the third pose here, over here, this is the fourth pose. In this way you can select all the molecules. So the binding pose that we require now is the first pose, which has better binding affinity. Okay, so go ahead and unclick everything. I think you don't need all the poses in order to visualize, in order to see the interaction. So go ahead and click this one, only this particular pose. You are interested in number 1 pose. You select this, and if you go here and if you directly drag the receptor file into the ChimeraX window, it will open the receptor file, and this is the first binding pose of your chemical compound or drug molecule added to the receptor, interaction docked to the receptor. So you can analyze the interaction by using this particular feature. This is only for the tutorial purpose. I'm not going to do the analysis that has been done in the paper. So this is just an overview how to do it. Okay, and one more thing, that if you want to drag this particular interacted compound with the receptor, then click option and just click the left mouse button, and you can move it around like this. So if you want to make it in the center, just option and make it to the center. Okay, and you can see here, this is the receptor file that is assigned number 2. So if you click here, the receptor will be selected, and if you click this one, the ligand file will be selected. So this is the tree window in which you can explore all your binding poses. There is another option. If you go to the select and residues, you will see that unknown residues will be designated a name called UNL or LIG. It depends. If you go to the PDBQT file over here and open it, text editor, I like Sublime Text, so which is very good, and you can see that the name convention of this particular ligand is UNL. So that's why if you select residues, you will find UNL. That means this particular ligand file has been selected. Since you have shown here the 20 different poses, all the 20 different poses have been selected over here. So you don't want everything to be selected, you just want the first binding pose to be selected. Okay, so here there is another option. If you go to the molecular if you go to the home tab, there you have the sphere option that will make it looks like a CPK model. If you want stick model, it will be like stick model, which is a default one, and if you want ball and stick model, it will be assigned ball and stick model. Okay, if you select something and you want it in a zoomed version, then go to the action tab and view. Now you will see that it will be zoomed, and the rest of the molecules, rest of the receptor molecules will be faded out. Okay, only this particular selected drug compound or the receptor will be highlighted. Okay, and there are other options in which you can change the background color to white, which is good for publication quality figures, and there is black option. This is for analyzing, but this is for publication quality figures. Okay, and there are three options for lighting. So one is simple, soft, and full. So if you click on simple, so this is the simplest view of the ChimeraX, which is the default one. If you go to the full, it will be a little bit more vivid, and this can be utilized for publication quality figures. And there is another option called soft, which will make it a little bit more transparent, and visually it looks absolutely perfect. I mean, absolutely elegant, this particular view. Okay, and the atoms are colored based on the UCSF Chimera default atom type coloring, which is the red one is oxygen. If you hover the mouse over here, you will see it will be assigned the name oxygen, and the color will be assigned automatically to the atoms that are designated, that is O, and this will be nitrogen, you will see N, and this will be hydrogen, which is white. The gray color are carbon atoms, and this one is fluorine, because the molecule that we have chosen has a functional group in which fluorine is attached. Okay, so this is how you can select the specific binding pose along with the receptor, and you can visualize it by using UCSF ChimeraX. And one more thing that you have noticed, that when you select, it will be in green colored selection. So if you don't want this green colored selection in your publication, just go here, select, and clear. Okay, it will not be selected anymore. Okay, or otherwise click here and untick the selected version. Okay, now moving forward, now we go for the molecular display. So if you unselect this and click on action and view, it will be automatically reset to its original view, which is centered one. Okay, so now you want to select this particular molecule. Okay, let me show you another option, that if you select the receptor file, okay, and if you click on this is the cartoon version. If you click this, so the cartoon version of the receptor file will be shown, and if you hide it, everything will be hidden. Okay, and if you want to show the atoms, the side chain atoms associated with all the residues, then you click on show, so it will show all the side chain residues, I mean the whole chemical structure with the backbone C alpha, okay, the carbon alpha, and these are the side chains. So this looks quite messy. When you try to publish this in your publication, then it looks quite messy. So one of the best ways is to hide it. So if you click because you have selected the receptor, if you hide, only the side chain residues will be hidden. Okay, now and there is another option, another tool. If you go to molecule display, and if you have selected the receptor, and if you click surfaces show, so the whole surface will be shown for the receptor protein, and this is your ligand file. Okay, so if you unclick this, so this looks pretty good for the publication, that you can show the binding pose and the clefts, and the clefts are shown like this, which is really good. And if you select this and click on action view, so it will be shown. Oh wow, this is fantastic. I mean, ChimeraX has been designed in such a way that it looks so perfect. You don't have to do it manually by setting the colors and setting the shades, or that you do mostly by using UCSF Chimera. I have made one video how to make publication quality figures by using UCSF Chimera, but UCSF ChimeraX has all the features that within just one click you can make good quality figures. Okay, so let me unselect that, and action view. So this is the original form. Okay, and then if you select the ligand and if you go over here the H bonds, if you click this, so automatically it will create different types of hydrogen bonds, which is pretty much not visible with the surface structure. So what you have to do is go here, click on this particular receptor file. Okay, and yeah, this particular receptor file, and hide the surface. So now if you can zoom in, so let me select the ligand and action view. If you zoom in, there will be a dotted lines, which is the hydrogen bond. Okay, if you hover the mouse over here, you can see that this particular ligand, the oxygen atom, is interacting with serine 158. Okay, so this is the serine residue. It is interacting with the serine residue and forming a hydrogen bond. Okay, this is a conventional hydrogen bond. So I mean, this is how you can show in a publication format, and you can just make the aspect ratio and your visualization settings so that it will be visualized while you take a snapshot of this particular figure and publish in your and put it in your publication. So this is a quite nice view, I would say, and you can click on option left mouse and you can just drag and see where exactly you can see the hydrogen bonds and different atoms of your ligand molecule and the receptor molecule. Since you have selected the action view command, so everything behind except this particular setting, everything behind will be shaded like this. And if you rotate and just adjust the settings in order to get a better view, and you can take a snapshot of this and publish this. So before taking a snapshot of this, you just have to unselect the ligand. So this will be unselected, the green shaded area will be deleted, and this you can take a snapshot of it. So how to take a snapshot of this publication quality is go to Home tab and click here, snapshot. It will take some time, and if you go to the desktop, I think it will be saved in the desktop, and you can see that it is saved as PNG, and you can see the quality of the figure is really good. So you don't have to do this POV, or there are some rendering options in PyMol and UCSF Chimera, but not in ChimeraX. ChimeraX has many features that it gives you a better quality figure. So this is one option that you can save a picture of this, a snapshot of this. Another option is go to file and save. I think yes, there will be option called PNG image, and you have TIFF image, especially for the PowerPoint presentation. PNG is quite useful for Keynote, I would say, and TIFF image is for PowerPoint presentation. And there are many different types of options, you can explore it, but I recommend, what I do mostly is save as PNG image or TIFF image, and then I proceed to make some quality figures. Okay, this is how you can save a picture of it. And if you want a video of this particular interaction video, if you want to make a video, just click over here and make spin movie. So it will automatically spin the movie and capture different frames until I think 180 frames will be captured. That will be the whole circular, covering all the aspects of the ligand and a full rotation, I would say. So You can see here that it is capturing 90, 100, it will go until 180. So if you click spin movie, it will automatically be saved in your desktop. So this is how you create movie. And UCSF Chimera, it was quite hard because you have to explore many different options in order to make a video, but here in ChimeraX it is as simple as that. It makes life easier. So you will see here movies saved to your desktop as movie.mp4. If you go here and just see how good it is, and this will be given as a supplementary file so that reviewer can understand what is the whole view, 3D view. Generally when you do a snapshot like this, if you take a snapshot like this, only one person, only one face has been visualized. Of course this is a 3D structure, no doubt in that, but still, if you want from all other sides, you want to see how it is interacting and what are the nearby residues, you can this one, that this is how the atoms are arranged and this is how the interaction is happening. Yeah, so let's move forward and make some adjustment to this particular hydrogen bonds. So if you go to the right mouse section over here, there are many different options. You can play around, but what are the essential tools that are required for a publication, that only I'm going to focus. So if you need the distance from this one to this one, if you hover the mouse over here, you can see that the distance is written over here, 2.265, and how to show it on your particular image. So go here in the distance tab, left click this and select the atom by using a right click, and select another atom which is required, which is interacting with this. You click the right click, okay, and then you will see that you have the distance written as 2.265. as you have seen over, when you hover the mouse, you will have the exact distance, that is 2.265 angstrom. Okay, so this is how you measure the distance of the hydrogen bond between different atoms. And if you want to click the residue name, which residue it is interacting with, so go over to any of the section of the residues and click on right click. So you will see that serine 158 has been written. If you want to name the ligand, you can just click on right click and you will see that UNL1 has been written, because in the PDB file the name of the ligand is UNL1. And if you want to show some side chain residues, or what are the different residues that are in the range of 5 angstrom or 4 angstrom, you can click here. You will see phenylalanine 112, and again you can click everywhere, whichever residues you require, and you will see that many different residues have been annotated just by right clicking it. Okay, so this particular view is quite good enough, so click on option and just make it center, and you will see the hydrogen bonds have been written over here. And if you want to move the level little bit closer to the interaction compound, so you just right click on this and you can move it around a little bit. Okay, so this is how you can label the residues, the vicinity of the interaction. You can label it, and if you need a closer view, just zoom in by clicking the middle mouse button and then option, and just put it right at the center so that it will be good for the publication quality figure. Okay, so unclick this one and then go to the home section, and if you want side chain residues also to be visible on your publication figure, then click on this one, the receptor, and click on show. So it will show different types of side chain residues, but it's quite messy, I would say. Only you have to show some of the residues. I mean, the ligand where it is interacting, it has already shown side chain residues and what atom it is interacting with. So you don't have to make a messy figure. You just have to hide it, and then click on serine by clicking particularly the serine residues by using control and then left click. And if you want to show this, I think the interaction, here, if you click on the hydrogen bonds and then click show, I think yes, it is being selected, but I think all the residues have been labeled. So how you want to do is just have to unselect this and click on hide on everything, select the ligand, click on show. Okay, and if you go to the molecular display and if you click on hydrogen bonds, it will be selected. Yes. So again, the original view can be you can bring it down by using the molecular display hydrogen bonds, and then you can see the distance, and because you have already labeled it, so you have everything written up here. Then go here, select and clear. And if you want a movie of that, and if you want to take a snapshot, you just take a snapshot at a better angle, I would say. Yeah, this is good. So take a snapshot. This is you save PNG image. Okay, then you click on the spin movie option, and it will capture again 180 frames, and then it will save as a video. Regarding the PNG image, you see now the labels, the hydrogen bonds, and all the protein residues which are under the vicinity of maybe 4 angstrom or maybe within 5 angstrom of the interaction compound that you have taken. So it's good to have some labels and the hydrogen bond distance, and this is quite a good image for publication purposes. And now I think the movie has been saved, but if you go to the first movie, there were no labels and you only have the drug compound and the receptor interaction, and this is good for supplementary purposes. But if you click on the movie 2, you see all the labels have been labeled, and yeah, you get more insights on the interaction profile. So yeah, so let's get some more insights from the interaction profile. That is, if you want to select the atoms, you know that hydrogens are labeled white, oxygens are labeled red color, nitrogens are labeled blue color, and carbons are gray color. Okay, so if you want to name the atoms also, press shift control and then left click button and go to the action tab, label and atoms, and then you can choose either name or element. Okay, if you choose element, then I think yes, the elements have been labeled. That hydrogen from serine 158 and oxygen from the drug compound, they are involved in the interaction profile. And you can also select some more atoms if you need, and you can also select the action label and name the elements accordingly. And if you want to save the movie, and you can also save by using the spin movie option, and it will give you a better insight or better perspective of your interaction profile. Then you take a snapshot. You can create a spin movie and recenter it to get a better angle, so that people can see in it, see the interaction in your publication, and they can understand. And we have three options called full lighting, soft, and simple. You can click on full lighting. Soft, this is what I like the most because it creates some vivid or elegant images. Even the simple is good enough for publication, so that it gives you more perspective, but I think the soft option is really good. It clears the background, and with a white background it generates a really a good publication quality image. So yeah, I think you can make such kind of interactions profile publication quality images from your docking results, and I think these images are good enough for publication quality. And one more important thing is that there are three main options in UCSF ChimeraX, that are home, molecule display, and the right mouse button. These three functions you require to analyze and to use it for your publication. Okay, I think this is all about UCSF Chimera. I'll see you in my next video with the Discovery Studio Visualizer, how to analyze by using Discovery Studio Visualizer. Welcome back to my channel. In this video, we are going to see how to make publication quality figures, and the visualization part will be covered today by using ChimeraX, Discovery Studio Visualizer, and there is a new software which is called Origin, in which you can plot 2D diagrams by using the numerical values that you have obtained from VINA results. So today we are going to cover these three softwares and how to make a publication quality figure. And rest of the softwares that have been listed over here, like Lig++ or the Python environment by using Miko, Anaconda, so these will be covered in my next video. So stay tuned for my upcoming videos, which will be very beneficial to run VINA by using Python coding rather than using different types of softwares. There are many tools which are implemented in Anaconda, so those will be covered in my next video. So basically today we are going to focus on Discovery Studio Visualizer, ChimeraX, and Origin, and of course how to make publication quality figures by using either PowerPoint or Keynote. So yeah, let's get started. So here the VINA results are here. So we have done blind docking and we have done the targeted docking. In the previous video I have shown how to analyze by using ChimeraX, how to see the interaction between the drug molecule and the receptor molecule, and how to plot it, how to make movies, how to take the snapshots by using ChimeraX. So today we are basically covering the Discovery Studio Visualizer, which is a Windows based software and Linux based software as well, but it doesn't work in Mac version. So that's why for the Mac people, they have to install this parallel software where you can use Windows operating system inside the Mac. So let's open the Discovery Studio client. This is a 2021 version. I think the new version has been released already from the Discovery Studio from the BioVia website. You can go there and download the Discovery Studio Visualizer. So this is the interface of Discovery Studio. For any receptor ligand interaction or any docking results, the most important tool in this particular visualizer is receptor ligand interactions, where you have to define the receptor, you have to define the ligand, and there will be ligand interactions, and you can visualize by using 2D diagrams. So how to do it? The good thing about the Discovery Studio Visualizer is that you don't have to go here, file, open, and you open the file. So there is a new option that is called drag and drop over here. So you go to the results of this blind docking, and you if you want to analyze the first binding pose of your drug molecule, you just drag the PDBQT file, and these files have been, there are 20 modes, and these files have been generated by using Vina_split. So you go to the first pose and just drag over here, and you see that the drug molecule, the binding pose number 1 has been opened in the interface of DS Visualizer. Okay. The next thing what you want to do is you have to drag the receptor file as well. So you drag the receptor file into the DS Visualizer window. You won't see the structure in this particular DS Visualizer window. Rather, you have a hierarchical tree in which you have your drug molecules, your receptor molecules. So the first thing what we have to do is we have to define the receptor, define the ligand, and then we do the ligand interaction. So how to do that? So this is your receptor file. So if you want to rename it, you can rename it as drug, and you can rename the receptor file as a receptor. Just double click on that and it will be renamed. So now you don't have any confusion that which is the receptor one and which is the drug molecule. Basically the drug molecules, they have heteroatoms in this, and receptor molecules, they have different types of chains. In this case you have chain A. Okay. So first thing what you have to define is your drug molecules. So go here, click on chain and define the ligand. Okay. Your current ligand has been selected. So this is your drug molecule, and this has been selected, or this has been defined, and UNL1, this is your name of the drug molecule, which is encoded in your PDB file if you open it by using text editor, and if you open the PDBQT file in the text editor, you will find the naming convention of the drug molecule is UNL. So it will be selected, current ligand. Okay. Now you have to define the receptor. So what you have to click is go to the receptor option and define the receptor molecule. Okay. Now the receptor molecule has been selected, and you can see that there is a new file that has been created which is called a receptor, SPD receptor. So now you have defined both the receptor and the ligand. Next, go to the ligand interaction tab and click on ligand interaction. So you will see that the receptor molecule and the interaction between the drug molecule has been, defined in the visualizer window. You see the different types of green lines, different types of I think teal green lines. So if you right click button and you can rotate this. If you are using Apple mouse, the right click option helps you to rotate the molecule. I recommend everyone to use the conventional mouse, because Windows platform, they do have different types of buttons, like of course in the Mac mouse there are left and right buttons, but for the scrolling option to zoom in and zoom out, the conventional mouse works pretty well. So I recommend everyone to use the conventional mouse rather than Mac mouse. Okay, so now you can see that your receptor ligand interactions has been depicted over here. And if you want to see what type of bonds these green colored and teal colored have been depicted, so what you have to do is go here and click on show types. So if you click on show types, then there you have all the bonds that are defined. So these are basically the green lines are the conventional hydrogen bonds, and you can see that we have fluorine in our drug molecule, so they have some kind of interactions with I think this is in between the ligand, but with the receptor molecule they have mostly conventional hydrogen bonding. So this is quite tricky to visualize each and every bond, so that's why DS Visualizer has been introduced to plot 2D diagrams, which are much more useful. And if you want to see the distance between the bonds, just click on show distance and it will show that this particular bond has 2.51 angstrom, this particular bond has 2.27 interactions. So the interaction that has been shown over here is a little bit confusing, that you can't see all the interactions at once and it is overlapping with each other. So what you have to do is go to the show 2D diagram over here and click show 2D diagram. So a 2D plot diagram will be generated. So this is much more clear, and even the receptor molecule which are in sticks, in ball and stick form, they don't have the labels defined. So 2D diagram helps you to define the ligands as well. So in the first glance you will see that your drug molecule is in a stick model, and this is pretty good for analysis, but for the publication quality figure you have to modify a little bit. So go here and click on right click. Okay, you have this 2D plot, different types of options. Okay, the number one is the general one, the background color is white. You have to show the legends. Of course you have to show the legends, otherwise that might be very hard to identify the interactions. So in here you will see you have a green line, those are conventional hydrogen bonds, and there are some carbon hydrogen bonds also, which are not quite visible, but mostly the conventional hydrogen bonds are visible over here. And you can define your font and scale with the different fonts, like here the default is Calibri, and you don't have to change the size of the font or the scaling parameter. If you change it, then it will be a little bit overlap. Just click on apply and you will see that some of the labels are overlapped. So stick with the default options. You don't have to change anything. So if you go to the second option, that is called atom, this is your drug molecule. So these are basically in the line format. This is the default one. If you need ball and stick, you just click on ball and stick and click on apply. You will see a nice view of your ligand, but the labels are not defined. Like these atoms, you don't know what these atoms are. So for better interaction visibility and for the better understanding for the layman people as well, if you want to label the atoms, then you can go to the disk shape and click on apply. You will see now, except the carbon, each and every atom has been labeled. Like the red one are oxygen, the blue one are nitrogen, and the teal color is fluorine atom. So this is quite useful, the disk shape, which will give better biological insights and your interaction profile as well. So I recommend everyone to go for the disk shape rather than ball and stick model. Of course, if your atoms, you know what type of atoms are there apart from all the different types of elements that are present, then you can go with the ball and stick model. Otherwise, go for the disk model. This gives a better visualization perspective. Okay. Then we go to the residue tab, where all these residues are the default is ball shaped. You stick with that. I mean, this is quite useful. Otherwise, go for the disk option. You will see a border line has been defined. So that's the only change, but you don't have to change much on the residue size. You can change the ball size as well. So if you click on 1.00 and see that it is scaled accordingly, which is good for visualization purpose also. If you plot it, I mean, if you copy and paste it in Keynote or PowerPoint, this will be a little bit enhanced or visualized at a defined scale. Okay. The next option is the interaction. You go here. In here, you will see different types of interactions like hydrogen bonds, electrostatic bonds, pi charge. If you have an interaction with the alkyl bonds or the pi charge or sigma, you will see here. This is the default one. So whatever the interactions that are imposed by the drug with the receptor, that will be shown here. In here, only we have two, and that is conventional hydrogen bond and carbon hydrogen bond. So nothing much, there are no further option to introduce any of the pi charge or non-classical, hydrophobic bonds or alkyl bonds over here. So it will automatically generate different types of bonds if your drug molecule has many different types of interactions. The next thing that we want to show is show distance. So if you click on show distance, it will show the distance in angstroms. So this interaction is particularly 2.73 angstrom. Okay. So these are the options that you can modify or you can adjust based on your needs. These are the basic options. So and the next thing that you have to do is copy this particular window, show 2D diagram, and I'm going to open a Keynote, a PowerPoint tab, and just paste it over here. When you paste the particular 2D diagram interaction, you see that the resolution of this particular 2D diagram has been retained. So you don't have to worry about anything. You don't have to modify anything. At last, I'm going to show you how to make it more vivid and elegant for a publication quality figure. For now, I'm just pasting the 2D diagram with it. So now you go and close it, and I have shown in my Science Advances paper that how you can make 3D interaction plot by using different types of options that are available on DS Visualizer with the hydrogen bond, with the aromaticity, or with the solvent accessible surface area. So if you want to generate these kind of diagrams, so this can be done by using Discovery Studio Visualizer. So if you want to define the receptor surface of your drug molecule interacting with the receptor, you just have to go here and click on edge bond, because this is what you are interested in. At first you won't see anything, any surfaces on it. So what you have to go is go to the script section and click on ligand interaction over here, and then create surface around ligand. Okay. If you click that, so you will see that a lot of receptor molecules, receptor residues has been defined to create the surface around the ligand. So if you see that in here, your drug molecule is interacting with a lot of donor atoms rather than acceptor atoms. Okay. So in this way, you can plot different types of receptor surfaces in which how your drug molecule is interacting with, and you can see that if you change the perspective of your bonding patterns or the visualization pattern. You can see a lot of adjustments. It happens. I mean, when it is interacting with donor atoms over here, they are colored in purple, and if there are any acceptor atoms, that will be colored as green. The reason why it is not rendering properly, because I'm using Windows inside the Mac, so it takes a lot of processor or CPUs. That's why it's a little bit lagging. So I recommend everyone to, if you are using Discovery Studio Visualizer, go with the Windows platform and create these kind of surfaces. And if you want a publication quality figure, then if you want the background to be white, go here in the scripts visualization section and click here publication quality. So everything, the background will be white and your results are a little bit vivid. This is in case of hydrogen bonds that you have three defined colors. One is green, one is purple, and one is in medium, that is white. So if you want solvent accessible surface area, then you see that everything is blue. That means it has high solvent accessible surface area. And if you want hydrophobicity to be analyzed, then you can click on all these different options to get different types of plots. So mostly if you have more conventional hydrogen bonds, then you go for the hydrogen bonds. If you want to analyze more deeply and to gain more insight, then you click on charge, click on ionizability, click on SAS, and all these options can be explored and can be plotted accordingly. So I'm going with the H bond atoms, and for this I'm copying this particular file, this particular picture, and I'm pasting it over here. Later on I will show you how to modify this, how to make arrangements to create a publication quality figure. I'm just keeping it for now. Okay, so this is how you make 2D plots and 3D plots using the receptor surface, using the ligand interactions, and your 2D diagrams. Okay, so if you click on the receptor surface first and you create a surface around the ligand, so you have many more atoms that have been defined over here. As you can see, there are many more residues from the receptor molecules which are in the vicinity of the drug molecule has been defined over. I mean, this is also good, but it creates a lot of confusion that what type of interactions that we are looking for, and what are the particular residues that is only interact with the ligand molecule. That is the most important thing. So you define the ligand, define the receptor, click on ligand interactions, and directly go to the show 2D diagram. That will define only the residues that is interacting with the drug molecule. If you click on the receptor surface and then click on show 2D diagram, it will show many more receptor residues, and I think you can see that a new interaction has been introduced that is called van der Waals forces. So in this way, I mean, it depends on you how you want to analyze, and this is the basic analysis platform. And the next most important thing is that you have a text of all the interactions that has been shown over here. You have a particular window in which everything has been defined. So if you go to the molecule, this is your drug molecule, this is your receptor molecule, these are chains of the drug molecule, these are the residues, atoms that has been defined for the drug molecule. And if you go to the amino acid chain, you have A chain, and amino acids, you have all the amino acids with different types of pK values, hydrophobicity, So for gaining deeper insight to a particular drug molecule interactions, these options are really useful, and you can see different types of bonding patterns that has been described over here. You can find it in here, and you can just copy paste these and put it in an Excel file, which is much more useful, I would say. Okay, so I think yes, you can just copy paste these, copy, and I think you can export it as well. No, I think you can copy this and open Excel file and just paste it over here. So you have this defined interactions that is in between the drug molecule and the receptor molecule, and you can give this directly for publications as well. Yeah, you can see that this is interaction with aspartic acid 153 with the atoms OD2, and this is a ligand non-bond monitor. This is the hydrogen bond, conventional hydrogen bond. The distance is 2.5 angstrom, and the labels are defined over here. You can just copy paste, or maybe you can just write it down. So relevant columns, you can write it down, and you can give this particular file as a supplementary, or maybe in your manuscript file. Okay, so this is how you basically utilize Discovery Studio Visualizer to see receptor ligand interactions. This is a very good software. I recommend everyone to use DS Visualizer so that you get more insights into the interaction profile. Just copy the 2D diagram, copy the 3D surface diagram, and you can plot it for a publication quality figure. Okay, so this is all about DS Visualizer and how to use it, how to analyze the V9 results using Discovery Studio Visualizer. Okay. The next thing that I'm going to show is how to use the origin plot. How to make 2D diagram plot for your V9 results. The numerical values that you got from V9 results. Suppose in this case you have a log file where your binding modes, and this is your binding affinity. So how to plot this? By using origin, and you might have known about different line plots or different column views. People generally use column view for receptor ligand interactions. So rather than that, I recommend everyone to go for box plots. So these are the defined modes. You just copy this and paste it over here, and I think the next thing that you want to do is copy the binding affinity values over here. Sorry. And while using Origin, because Origin is a Windows-based software, it is not in Mac. So I generally suggest everyone to use the conventional mouse rather than Apple mouse, because sometimes it's annoying to use Apple mouse as well. So you define it like binding modes, and in here you define binding affinity, label it again. Affinity, this is in kilocalories per mole. Okay, and you delete this column because you don't need this column. So you have your binding modes over here, you have your binding affinity in this particular column. So select X and Y and go to the right click and plot line and symbol. Okay, this is one way in which you can plot your Autodoc VNAR results. So basically the labels are inverted, so I'm going to change it from this. The scaling pattern has to be reversed, because this is a negative less, and this is 4.25, I guess. and you can see that the binding modes for a better binding affinity has been defined at top. That is minus 6.5 over here, or 6.3 here, and this is the least binding affinity that you get under 20th binding pose. So it has to be defined like this, because the higher the negative value, the more the negative value, the high is the binding affinity. This is what we get from Autodoc Vena results. The more the negative value, the highest is the binding affinity. So generally what people do is they just give only the first binding mode results, which is good in the sense that you have a defined interactions, or you have a defined binding affinity for that. But if you want to show the 20 different poses and how the binding affinity scores has been distributed along with different binding poses, so this is one of the best way to plot, so that reviewer can understand that okay, you have done 20 different poses and you have a scale score from the highest binding affinity to the least binding affinity. Okay, this is one way that you can plot. Another way that you can plot is go to the statistics in here and click on box chart. So this is again another way that you define your Autodoc Vena scores by using box plot. So most of this is the mean, and if you have any outliers, that will be in a diamond shape, and this is the median line. So you have 20 different poses. So what is the mean and what is the median of these interaction scores that you have obtained from Autodoc Vena? That is very important. And what is the highest one? What is the highest one? What is the least one? So that you can also define over here. The box plot is much more useful if you have several different binding scores. 4.25. I'm just changing the scales so that this is your highest and the first binding pose. This is your least binding pose, which has low binding affinity. And most of the scores are distributed under this range, that is minus 5.5, I would say, or minus 5.7 to I think 4.8. So most of the interactions are under this. This is your highest interaction. This is your least binding affinity scores. I'm going to show you another way that you can plot many different Vena scores. So in this case, if you click on add new column, and I have created a test file to see how different, if you have drug 1, drug 2 to drug 11 or many more, and you have 20 different poses. So you click on this and just paste it over here. So you have all the molecules that are defined. Okay, the binding scores have been defined, and these are your drug molecules. Okay, click in here and paste the labels. Suppose I name it as drug. Okay, drug 1, drug 2, drug 3, and these are your binding modes. So if you create a nice box plot on this, you select all the Y columns, go to the plot and statistics and your box chart. So you can see a nice view of the box plot can be. I'm just deleting this to show you how it looks, and you can see that these are your drug molecules. You can just change the labels and rotate it to, I think, 60 degrees or something. Yeah, let me rotate it to 60. Yeah, now you can see this is your first drug molecule, and this is drug 1, drug 2, drug 3, drug 4. So rather than plotting it by using line and plot, so If you want to plot using line and compare it with box plot, the box plot looks much more, vivid. You can see. So in comparison to this plot, the line and symbol plot to box plot, the box plot looks more vivid and it has more insight rather than confusing line and symbol plot. So I recommend everyone to go with the box plot. Suppose you have this interaction, you just copy graph as a picture and save it on your paste it in your Keynote or Discovery in your PowerPoint. Okay, now you have 3 diagrams. One is 2D plot, one is surface one. You have your scores as well. Okay, so this is all about Origin. You can explore many different options in Origin. You can different the plots in a very schematic way, very a good presentation that you can make by exploring different options in Origin plot. So yeah, if you have many different VNAV scores, then go for the box plot, which is good. Do not go for the line plot, which is really confusing. Yeah, this is all about Origin. Okay, so let me just close it, and we are done with the Windows platform right now. Okay, so this is your Keynote file. Now the next thing is how you get a nice view plot from Chimera. Okay, so I'm going with the blind docking option. So you open the drug molecule in ChimeraX, not UCSF Chimera, but ChimeraX, because it directly can open the PDBQT files in the window. Okay, so this is your drug molecule PDBQT file, and here likewise in Discovery Studio visualize your drag and drop. Here also you can drag and drop, and you can see your receptor and the drug molecule has been defined. How to do the analysis I have shown in my previous video, but here in this particular video I'm going to show you how to plot a nice view from UCSF ChimeraX. So the first thing that you do is you click on white background, because black backgrounds are not accepted in any publication. And the first thing that you want to do is create a surface around your protein. So what you have to do is go to select, change, and your receptor molecule. Go here in the molecule display and click on show here in surface. So you have your surface defined, and this is the default blue colored surface will be presented when you click on surfaces and show. Okay, but this is not this doesn't look so elegant or vivid. So what you want to do is, generally receptors have different types of coils, helix, and strand structures. I mean beta strands. So you want to color them and make it more elegant and vivid. So how to do that? So go to the select option, go to the structure, and go here secondary structure. This is only for the ChimeraX. For UCSF Chimera it has other options, but this is only for the ChimeraX. So go to the second structure and click on coil. Okay, now the coil regions have been selected. Basically the coil regions, they are labeled, they are colored in light gray or whitish background. So I would go with the light gray background for now. Okay, and if you click on the structures with the helix, the helix region has been selected. Now we go for the action color, and I color it as light sea green. Okay, now you go to your strand and action, color it as maybe cornflower blue or blue. I go with cornflower blue. Now you have your strand, your helix, and your coil region, and this is your ligand molecule. Okay, so go to the ligand, go to the residue, select the ligand, and click on ball and stick model. You can go with the spear as well, but it doesn't look that much good for a publication quality, so you go with the ball and stick model. Okay, now clear everything, and here there are three options that I have shown in my previous video: simple, soft, and full. If you go to the simple, this is the simplest view, which is the default one for UCSF ChimeraX. If you go to the full view, it gives you more shades. Okay, if you go to the soft view, yeah, it creates a publication quality figure. Now you can see that your ligand molecule and your receptor molecules, when you copy this and paste it in your Keynote file or PowerPoint, it looks much more, a very nice view of your interaction can be, presented there. So I'm going to click here on snapshot, and it will create an image, and we can see later on how to, make a nice presentation by using Keynote. Because I'm using Mac, so I'm using Keynote. You can do this in your PowerPoint as well. Okay, now I have created a snapshot of it, and if you click on the residues and you select the ligand and click on view, you will see a zoomed view of your ligand molecule and your receptor molecule. So we go ahead and select clear and get a snapshot out of it. This is another snapshot of your presentation. It takes some time because this is with the surface and your ligand molecule. Yeah, now it is created. It is saved on your desktop or maybe in some other location. Okay, so you get this nice view. But if you go here, residue, chains, and go to the molecule display and click hide the surface. You don't need the surface. Now we need the interaction profile between the receptor molecule and your ligand interaction that I have shown in my previous video, but I'm doing it for the sake of presentation. Go to the select residues, UNL, and hydrogen bonds over here. Now you have one defined hydrogen bonds. Go to the right mouse, label, and you just click on the labels over here. Okay, so this particular view also you can make for your presentation. Okay, and distance, so we have this, yeah, this is your distance. Okay, now go to home and just click a nice view. So this is a particular view that you require for your presentation. Make it center a little bit. Okay, then just clear it and take a snapshot out of it. So now you have 3 snapshots from ChimeraX, 2 snapshots from Discovery Studio, and 1 from Origin. Okay, now you close this window. Now we should move into the Keynote presentation, how you make the presentation. The first thing that for the Keynote, any presentation for publication quality figure, that you have to make it to A4 size. So in PowerPoint you have points, not the centimeters or not the mm, but you have points, which is 595 into 842 points. So what you have to do is go here in the document section, go to the slide size. You have default standard, you have default widescreen. Go to the custom slide size and make it to 519 to 842. Okay, now you have your A4 size. Okay, and in here you have to create your presentation. I think 842, yes, this is your A4 size figure. So go to the desktop and you have 3 different options. So number 1 is your surface view of your receptor and ligand. This is the zoomed view of your surface and ligand. and the third one is the interaction view of your surface in Ligand. You just grab everything from different softwares and paste it in the Keynote. So I'm just going to show you how to make these plots look much more vivid and elegant for a publication file. So if you paste this, so you can see that there are a lot of white spaces. If you don't have any labels in your diagram, then go to the format option, go to the image option and click on instant alpha and click on the white spaces. So it will automatically delete all the white spaces and your only the defined region of your molecule will be plotted over here. So the white spaces will be removed from it. Okay. The next diagram that is your zoomed view. So you click here and see there are a lot of white spaces. So go to instant alpha and click on the white spaces. So white spaces have been deleted. Now you have your surface and you have your zoomed view of your ligand and protein interactions. The next thing is your interaction profile. So here you have defined labels over here. So if you click on instant alpha and do this, your labels will be a little bit faded away. So you don't have to do that. Just revert it. You don't just reset it. Okay, because you need your labels to be visible to to the readers of your scientific article. So you just go here, double click, left mouse double click, and you can see crop and scale option over here. So you just crop the white spaces from here if you don't need this. Okay, and you can just make it a little bit cropped so that you won't miss out so many residues, but only the white spaces will be removed. So this is your interaction plot right now. The next thing is the receptor. This is the hydrogen bond receptor. So go here, instant alpha, click on the white spaces, and you have your H bond interaction. The labels from Discovery Studio Visualizer, you can just take a screenshot and just paste it over here, or you define it by yourself by making a small little square box or rectangle box and define H bond donors and H bond acceptors. Okay, so this is another view. Now you have your 2D plot. Okay, and you can also do the same by removing the white spaces from here, and at last you have your 2D plot. So you remove the white space because here there are labels, so you don't have to click on instant alpha, so that it will the labels will be faded away. So you just go and crop and scale it. So now you have all the interaction profile for a particular drug molecule interacting with the receptor molecules. Okay, so now click on all the molecules. There is a nice option that you can enhance your image. So if you click on enhance, so it will be a little bit enhanced, and this is ready for publication quality figure. So if you click here and go for slideshow, and you see a nice presentation of your interaction, the drug molecule interaction with the receptor, and this is, I think, is ready for publication. And by creating such diagrams, you can explore many different options in ChimeraX, Discovery Studio, Origin, and you can plot very nice figures from your analysis or from your VNA results. Okay, so this is how it looks, and if you want to save it, just go to export and PDF file. This is only for the Mac users, because if you save the PNG image of this particular file, the resolution will be reduced. So you have to save it in PDF first. Okay, desktop, and let me name it as presentation. Okay, now a PDF file will be created, and if you want to save it, presentation. Okay. So a PDF file will be created in which the resolution is retained, and from here, when you open the PDF file, then you have the export option. You go here and click on PNG with 300 resolution. This is the recommended resolution for any publication quality figure. Click here, make it 300 pixels per inch, and save it as PNG. So if you go to the desktop, it is saved in your desktop. So if you click on the PNG file, you see that the resolution has been retained. There are no low resolution files or formats. You can see the resolution is maintained over here. And if you open Word file, because generally most of the manuscripts are written in Word file, so if you click on a blank document and paste in here your PNG file, and you see the resolution is maintained, and you scale it to 100, and now you can see your publication quality figure. The defined labels are fine, your visualization or your picture looks very elegant and vivid, and I think you might go for high impact journals as well. And this is how you make publication quality figures by using 3 different softwares. Those are ChimeraX, Discovery Studio Visualizer for these 2, and your Origin for plotting 2D diagrams using the numerical values you obtain from Vina. So this is all about how to do molecular docking, how to present the molecular docking results in an elegant and vivid way. And in my next video, I'm going to show you how to do the Vina docking by using Python environment, by using Anaconda, Miko, Lig++, several other tools are there, but these are some of the basic analysis tools. You can do it for auto docking Vina only. Okay. So for now, it's done, and see you in my next video. Thank you very much.